Okay, epigenetics. What is this? This is a field now that is getting hot, hot, hot. It's so hot that you can't hold it in your hand without scalding and burning yourself. Anybody wants to do a paper? If you want to go into something that is a relatively new and extremely hot field, you can try something on this. There's a lot of cool stuff, and this is a wide open field. There's a lot of research. We know relatively little about it still, but we're beginning to realize more and more that it's extremely important. So let's take a look at a few key points about this. Okay, now what is epigenetics? If you want to define it, you could say, okay, if you want to make a definition of epigenetics, you could say it's generally, there are some exceptions, exactly how you define it, a little bit nebulous, but in general we're looking at fairly stable, fairly long-term changes in regulation of genes, in transcriptional regulation. Now, that's one thing. So, if you want to ramp up transcription of a gene big time, you can do these kinds of things. And keep it that way for a long, long, long time. This is one thing you can do. If you want to repress transcription of a given gene for the long term, this is what you can do. But the most interesting thing about epigenetics is some of these changes can be passed down to the daughter cells. They're heritable. So, for us, whatever epigenetic things mommy had may end up in you. Some of these things are transmissible from one generation to the next. Now, how that happens, we'll see a little bit later. Okay, so that's, you want to say, the capsule definition of epigenetics. Now, how and why does that work? There's a lot we don't know about, but we're realizing more and more importance about it. Okay, now, let's take a little look at something. One of the first discoveries about this came from an experiment that was done about 20 or 30 years ago. Okay, we take an egg cell, fertilize it, put a sperm in. And of course, you now merge things together to make a diploid genome, and you get normal development some of the time. Okay. Now, if you take an egg cell and activate the developmental program, they literally call it activating the egg, is the program for early development is stored in a series of transcription factors and other kinds of proteins and messenger RNAs in the cytoplasm of the egg. So the egg can support early development even without anything new being transcribed or produced. Okay, now, you do that and activate an egg that has a haploid egg nucleus in it, it will indeed start to develop. And it will divide a certain number of times and then all of a sudden arrest, stop, and break up. It's kind of like, you remember the old Roadrunner cartoons? How many times was the Roadrunner running after, or the coyote running after the Roadrunner down this desert road, and all of a sudden goes off this cliff, and he's running out, and there's thin air and nothing below him, right? And he looks down and sees half a mile of empty space, and then he plunges down. That's kind of like what we see here with a haploid nucleus. It starts undergoing the developmental program, and it realizes, oh crap, I'm missing something. What are you missing? You're missing that other haploid genome. And then you get developmental arrest. Okay. Let's do a little experiment here. Okay, we need a diploid genome for normal development. So, clearly if you put a sperm nucleus and an egg nucleus together, it happens. Now you can do an experiment where you inject the sperm nucleus into the egg, that activates the egg, you get the fusion of the nuclei, a diploid genome, you get normal development. But let's try something different. Let's make a diploid genome another way. We're going to take the egg, and then add in an egg nucleus. A nucleus from another egg, put it in there. They fuse together, now we got diploid genome, it's activated, and what happens? Not normal development, folks. Development proceeds, but it's highly abnormal. You find that the embryo is much smaller than normal and certain tissues are vastly underdeveloped. And some things like the egg membranes, the amnion, the chorion, stuff like that are vastly overdeveloped. So we have misdevelopment here. Some parts overdo it, some parts underdo it. So even though we have a diploid genome, we're not getting proper development by using two egg nuclei. Well, let's do another experiment here. Let's suck the egg nucleus out and put a sperm nucleus in here. And then we add another sperm. Once again, we generate a diploid genome, development proceeds, and lo and behold, it's again abnormal development. In this case, the embryo grows exceptionally large and fast, overdevelopment of certain tissues, underdevelopment of membranes like the chorion and the amnion, that kind of stuff, and basically development's all screwed up here. On the other hand, you take an egg nucleus, even suck the nucleus out of the original egg, put another egg nucleus in, put a sperm nucleus in, activate, you get normal development. So it's saying something. In order for development to proceed properly, it's not just having a diploid genome. You have to have a haploid genome from egg, a haploid genome from sperm for development to proceed. If you use two sperm or two egg nuclei, you got your diploid genome, but things go wrong. So the question is, how and why do things go wrong? Well, that leads to one of the aspects of epigenetics. When people start looking at various genes and later on genomes, they found something intriguing. They found that certain bases on DNA, in fact I think it's cytosine, let me just double check that, I think it's cytosine. Yes. Okay, they found what was called DNA methylation. DNA methylation means that there are methyl, that's the hydrophobic carbon and 3 hydrogens, that there are methyl groups attached, covalently attached to DNA, specifically the bases, the cytosines. Now, not all cytosines get methylated, but some do. These methyl groups are attached to the cytosines by enzymes called DNA methylases. And when people start looking at methylation of DNA, they would find that certain genes had methyl groups stuck at certain places. And generally, when you had DNA methylation, usually it meant that that gene is strong, the gene that is methylated is strongly repressed. In other words, transcription generally doesn't occur on genes that are highly methylated. Now, it's interesting, we've seen DNA methylation before in prokaryotic biology. Bacteria methylate certain parts of DNA, and that protects them. That makes that DNA unable to be attacked by the restriction enzymes they put out. Recall, restriction enzymes are enzymes that cut at specific short DNA sequences. Now, the idea behind bacteria putting out these restriction enzymes to begin with
is if a virus gets in, the biggest threat for bacteria are bacteria infecting viruses, what we call bacteriophages, literally bacteria eaters. Those are the nemesis. That's the Andromeda strain from bacteria. Well, if you're a bacteria, you're always at risk from bacteriophage viruses. So what they do is they produce these restriction enzymes. They recognize sequences and basically chop the viral DNA into little pieces, and that means that most bacteriophages are DNA viruses, and that means you just save yourself from a really nasty infection that would kill you rupture you in about 20 minutes. Okay, well, the problem is you've got those DNA sequences in your own genome, so you have to protect it. The methylation protects, the restriction enzymes cannot attack their sequences if some of the bases are methylated. So that's how it probably started in bacteria. But now, we're not so much dealing with bacteriophage problems here because they don't affect us, but we do have the methylation here. Generally, methylating DNA and genes and things like promoters and stuff like that, that generally represses transcription quite strongly. It shuts the gene off and it shuts it off long term. So that's one of the things, DNA methylation. But the thing that was astonishing about DNA methylation is you could take a cell and you look at the methylation pattern of a particular gene in that cell, or a set of genes or what have you. And then that cell divides. And lo and behold, the daughter cell has the same methylation pattern. So that raises the question, how can we can see how we replicate DNA passing the same set of genes to the two daughter cells. The question is, how can we pass this methylation pattern down to the next generation? And that's where we seem to have kind of a particular type of mechanism. Okay, and we'll see that in just one moment. Okay, so that's one kind of thing. Methylation patterns can be inherited. Now, you can also unmethylate DNA. There are, these are DNA transferases. It's the methyltransferases that actually add methyl groups to DNA. And then there are DNA methylases which remove them. So you can methylate DNA, but you can also remove methyl groups when we want to. So there's a whole slew of these things that add methyl groups to DNA, others they can remove. Now the question is, how do we get a heritability of a methylation pattern? Well, it turns out it's actually the methyltransferases that do this. The methyltransferases can bind to DNA. Obviously they have to methylate it, but they bind to methylated bases 1,000 times more powerfully than they bind to unmethylated bases. So what's going to happen here, a crude model here. Here is our DNA, the double helix. And what I'm going to do is I'm going to put a little methylation pattern here. Let's put M for a methylated base. So I'll put on this strand a methyl group here. On this strand, I'm going to put a single methyl group. Yes?
On your DNA methylase that you have circled, what is that word right there?
Removes methyl group. Sorry. Okay, that was it. Okay. Here is a little segment of double-stranded DNA with a methylation point. Now, how are we going to pass this down to the next strand? Let's separate the two strands apart at what we would call the replication fork, when we're actually separating the strands of DNA and synthesizing the new strands of DNA. I'm not going to draw the replication fork, but here we have it here. Okay, now, here again, it's a second base here that's methylated, 2, and here's another methylated one, and in this case, it's the third base that's methylated. Okay. Okay. Now, we're going to synthesize a new strand of DNA.
Now what's going to happen? This new strand here is going to be an exact duplicate of this strand. In fact, we'll call this strand A, this strand B. How about that? Now this strand here is going to be a duplicate of strand B. So let's call this strand B prime, and this sister over here is going to be a duplicate of A. We'll call this A prime. Okay. Now what's going to happen? Here these are physically spaced quite close together, actual three-dimensional thing. This would be kind of a replication for, the new strands are being formed over here. Okay, so they're physically quite close together. Now our methyltransferases, what they'll do is they will bind strongly over here, and they can reach over right to the comparable strand, that newly synthesizing strand on the other side of the replication fork, and add a methyl group here. Likewise, this one here, the transferase, we call T for transferase, the transferase reaches way over and puts a methyl group here. Now, we have a transferase over here. It's going to reach over and stick a methyl group here. Now this has to, obviously before the two strands of DNA are physically separated by a large amount, this has to happen very early on as we're synthesizing the DNA at the replication fork. These have to be quite close, physically close together. But the way that this thing is inherited is the methyltransferases bind to the methylated bases. They reach over and add methyl groups to the newly synthesizing strand that's its sister on the other side of the replication fork. And that way you can inherit the methylation pattern.